Assalamualaikum. My name is Rohma Tahir and I am the host for today representing Bioverse. So I would like to introduce Dr. Fatima first. Dr. Ambrin Fatima is actually an assistant University, Karachi, Pakistan. Her primary and intermediate aim is to analyze the families with genetic disorders all the way from the identification of causative gene variants to unravel pathophysiological mechanism with the perspective of therapeutic interventions. Dr. Fatma has more than 11 years of experience in human genetics with 6 years of international experience at the University of Copenhagen, Denmark and Uppsala University, Sweden. Dr. Fatima has extensive expertise in disease gene identification by using several next-generation sequencing platforms and disease modeling techniques such as iPSCs, gene editing by CRISPR-Cas9 technology, differentiation of iPSCs into different neuronal cell types, and CD cerebral organoids. I am very humbled and honored to having Dr. Amreen Fatma on our platform. I would invite her to start the webinar now. Thank you so much for being here, Dr. Fatma, and
Uh,
you can start.
Thank you so much for the introduction and for the invitation. So, I'm a human geneticist, and of course, I'm interested in stem cell based disease modeling. So, today I'm going to drive you quickly through some techniques or like the possibility of using stem cell for disease modeling and for different drug discovery as well as for the cell therapy. So, okay, so we'll start with the disease modeling. So what actually disease modeling is? It's just to way a way to see the pathophysiology of a disease. So how we see the pathophysiology. So if there is an inherited disease, so we can see that there is a mutation that is causing the disease. So we can see the disease phenotype, but how that disease progress and what actually disease effect in at the cellular level, so we cannot see. So so what we do to look on the pathophysiology of the disease, so we use different model system. So that could be animal model, that could be cell model. So in animal we have plenty of models. We use mice, we use zebrafish, we use fruit fly, and so, some others. And of course, they have some advantages because these are the in vivo systems. So what we look is inside the body, inside the living organism. But beside having certain advantages, there are a couple of disadvantages as well. Like, of course, mice cannot be human or zebrafish cannot be human. And if you place their common ancestor, they still can be like million years apart in the evolution. And of course, human are very complex. So we'll see in the next slides as well. So what other options we have to study human diseases? So we can use the postmortem tissue, we can use some cell lines, but of course, again, they do not provide us enough liberty to use, to study all the genetic disorder or the human disorder, I would say. So for that, we use stem cells. So we'll talk about those further, like what these stem cells and their uses are. So as I mentioned, that human is not mice. It is not zebrafish. It's actually very complex organism. So even if you look on the brain, I have in this slide you can see the brain of different organisms. So even though the brain of elephant and some other mammals is larger than human, but human brain actually still hold the record of containing more number of neurons, and it's one of the most complex brain. So you cannot really study the brain in animal models. So for that, we need a model that can actually tell us about the human pathophysiology. So for that, we will use stem cell, and how we'll use the stem cell, we'll go to that part later. But what stem cell is? So stem cell have two properties. So the one is that it can replicate. So it can actually make more copies of similar copies. So it have a potential to grow and to make more cells of its similar type. The other property that any stem cell have is that it have a potential to make a different cell type. So one is that it have the potential to regenerate, it have the potential to grow, and the other is that it can become any other cell type. So it can, like, the stem cell can become heart cell, muscle cell, kidney cell, or brain cell, or whatever cell that you are interested in. But where these stem cells are located in human, so if we talk broadly, there are two types of stem cells. So one is embryonic stem cell, and the other is adult stem cell. So the embryonic stem cells are basically early in the embryonic development. So it's at this stage it's called blastocyst. So it's actually 4 to 5 days old embryo with 50 to a little more cells. So the cells at this stage, at the blastocyst stage, they are called stem cell, and they actually have ability to make any cells of the body, because further they make germ layer like ectoderm, endoderm, and mesoderm, and then they can differentiate it into their derivatives. The other stem cell that we have in our body, there are some that are in the fetus, like the newborn. For example, you can find some stem cell from the cord blood. And then there is another, like human, we all have certain stem cell in our body. For example, in bone marrow, we have hematopoietic stem cell, we have mesenchymal stem cell in our body. So, but those cells are different from these cells because these can make any cell type. But for example, the cells in the bone marrow, they can actually make few cell types. So that's why they call multipotent. But the cell that can make any type, they can call pluripotent. So where the scientist was getting pluripotent stem cell, of course, the source was in vitro fertilized embryo. So basically, when there is a fertilization, and then of course they grow the embryo for a couple of days, and then after that, you can actually remove the cells through a surgery. So basically, you inject a needle and you take the cells out from the blastocyst stage from very early embryo, and then you can put the cell on in the culture. You provide them nutrients and the media that they require to grow, and once you have these cells, now these are ready to make any cell type you are interested in. So people have started using the embryonic stem cell from IVF embryos, in which or fertilized embryo, for the research purposes, for the disease modeling, as well as for drug discovery and these things. But there are certain limitations of using embryonic stem cell. So the first one is it's immunogenic. So for example, if you are taking from an embryo and you cannot put these cells into any other human, because of course the body will react against, and it will be basically there will be immune response from the body. The second is most of these stem cells, since they have the ability to make a new cell type, so they are actually carcinogenic. I mean, they can make tumor. They can actually, instead of treating a disease, you can make it worse. You can cause these can lead to the cancer. And the other thing is, of course, when you get from the inner cell mass, they require the microsurgery. And then again, you need embryos to get these. And of course, you destroy those embryos. So, so to overcome this limitation, so there was this scientist from Japan, Shiana Yamanaka, and his postdoc. So they actually came up with the discovery, and what they did. So they took normal cell, like skin cell. So what they did actually, they took the skin biopsy. So and of course from the biopsy, they actually grew the fibroblast, the skin cell, and they were trying to put some transcription factor, and they actually started with 24 transcription factor, but later they were able to use 4 transcription factor, and these 4 transcription factor, OCT4, SOX2, CMAC, and KLF4, they are also called Yamanaka factors. So they have the ability to make somatic cell or mature adult cell to back into stem cells. So we call them reprogramming. So basically, if you want to have cell similar to the embryonic stem cell from you, you can get it. It's like, for example, if I want to get my stem cell for you know for a certain purpose, it could be for the to treat a disease, it could be for the disease modeling or anything, but I just need any cell type, like adult cell type. So it can be from blood cells, it can be from even our urine have some cell types. So there are a couple of reports where they actually use the urine cell to make stem cells, and of course the skin biopsy. So these cells, of course, they also have potential to differentiate into different cell type, into all, practically, like theoretically all cell type of our body. So, and on this discovery, he got Nobel Prize in 2012, and along with the other scientists who work on somatic nuclear transfer. So, of course, these discoveries were decades apart, but they actually had a similar kind of work, but and they got Nobel. So we studied, now we talk about the embryonic stem cell and the adult cell that Yamanaka made. So how they are similar. So one is coming from zygote, one is coming from embryo, and of course from the blastocyst. The other is coming from normal cell of your body. And once they are in culture, so there is no difference. So they we call them pluripotent stem cells. So one is embryonic, another is induced pluripotent. So basically they are similar in their characteristics. So they can become any cell type. So you can differentiate into ectoderm, endoderm, mesoderm, any cell type of your body. So basically, this is a very busy slide, but here are the pluripotent stem cells. These could be embryonic stem cell, it could be induced pluripotent stem cell. So what you can actually do with these cell types, so you can make any cell type that you are interested in. You can make the derivatives of endoderm, you can make the mesoderm, you can make ectoderm derivative, and actually you can make, so theoretically, it is there that you can make the germ cell, you can make the egg and sperm in the lab. And of course, so there is a lot of debate on this one, that okay, if you can make sperm cell, you can make egg cell. So basically, you can give this opportunity of having baby to an infertile couple. So, but of course, it's still, there is a lot of research and needed on this side, but probably this is also around the corner. So I highlighted this here because, of course, I'm working mostly on the neurodevelopmental disorders. So, of course, you can make neurons. I will show you some of the neurons that I made in the lab from these stem cells. And then you can make different types of neuron. Like, for example, you can make GABAergic neuron, you can make interneuron, you can make dopaminergic, any cell type that you are interested in. So when we make these cells from, we make iPSCs, and then we differentiate those iPSCs into a cell type. So what we do with those cells? So there are a couple of possibilities. So one is that you make the cell. So now I will talk about this isogenic thing later in the slides. So then you make either neuron, muscle cell, heart cell, that disease that you are interested in. And there are some possibility of making 3D brain organoid. So once you have these cells, you study different disease mechanism by using transcriptomics, proteomics, electrophysiology, microscopy, that you can look on different pathways and different, pathophysiology of disease. So once you know this, then you actually this can give you clue for the drug development or personalized medicine, especially in the term of that we call pharmacogenetics, or like, tailor medicine. There are certain names. So when you know the some biomarkers or some targets, so you can actually study those targets, and that will ultimately can lead to the personalized medicine. So we can do the disease modeling, we can do the drug discovery, and the second thing that we can do actually is, so we can put these cells, we can differentiate these cells, and we can put these cells back into the patient. For example, if patient have a neurodegenerative disease in which the neurons are degrading, so then you can actually make the neurons outside and give the patient those neurons in case of Alzheimer, in case of ALS disease. So we'll actually talk about those later in the slides. But before we do these all wonderful things we do for disease modeling or drug discovery or even the regenerative medicine, we have to make sure that the line that we have made is actually an iPSC line. It is actually a stem cell line. So we characterize those by using the morphology. We go for the gene expression, some transcription factor that only express in stem cell, but not in adult or fibroblast or the cell that you actually took to make the iPSC. So you look for those marker, you do immunostainings, and then of course you also look for the karyotyping, or because these process when we make or the different reprogram somatic cells into iPSCs, that can actually cause some uh, chromosomal aberrations. So they can actually delete some chromosome or a part of chromosome. So we have to make sure that everything is intact and this is the right line. And of course, they have the capacity to freeze, thaw, and regrow. Plus, they should also have the capacity to make the the different cell type, to differentiate into different cell types. That's the ultimate goal. So how we do that? So for the differentiation, we usually make embryonic bodies. So embryonic bodies is when you put the cell in a culture and uh, they have this ability to make aggregates. It's a self organized aggregates. So they look like somehow this is like one of our experiment that we did on EBs, embryonic bodies. So basically, this stage represents the very early embryonic development. So these embryonic bodies have the cells from ectoderm, endoderm, and mesoderm. So if you are interested in looking for early development or events in the early development, so you can actually make EBs. And plus these EBs also tell us that the line that we have made have the potential to make, to differentiate further. So this is the line that we have generated. We have generated certain lines in the lab, but this was from the Down syndrome patient. So this is how the iPSCs colony look like. And then we did some staining or also some flow analysis to look that the cell that we have made are actually expressing the pluripotent markers. So there are a couple of markers, for example, SOX2 nano, and we have SSDA4, and we have certain others. So, and then of course, so here, this is an iPS line and this is an EB. So I mentioned that EB is actually represent ectoderm, endoderm, and mesodermal cell. So basically, this analysis called scorecard. So it have a marker from different germ layer and from the pluripotent stage. So here you can see that in iPSCs, we have these in negative values. So it's mean the iPSCs are not expressing these cells of ectoderm and endoderm and mesoderm. While when we make them into EBs, embryonic bodies, so they express the cell of ectoderm, endoderm, and mesoderm, while they don't express the cell of pluripotent stage. And also we look on the karyotype, if it's normal, if it's intact. Since this was the Down syndrome, so they had actually 3 copies. So when your line is ready, you now have checked everything that it have the chromosomes are intact, it have the potential to differentiate, it look fine, everything is intact. So now you are ready to go for the disease modeling. So how you actually go for the disease modeling? So we usually take in past we used to take cells from healthy individual and a patient, and then we make iPSCs from both, and then we make desired cell type. For example, in this case, if it's a neuronal phenotype, so we make the neuron, and then we study that how the neuron from the patient are different from the control. Are they shorter? Are they have less connection? Are they have some problem with the synapses? So these kind of things, and of course you can study their transcriptomics, proteomics, and further things. And ultimately you go for the disease drug discovery. But one problem that we have with this way is that, okay, for example, if you are studying a genetic disease, and I'm sure you are familiar that sometimes there is only one mutation. So out of 3 billion base pair in your genome, if there is a change in one base pair, that can lead to the disease. So, so basically, we all are different. So even the control sample that we take and the patient sample we take, they have a different genetic background. So the ultimately the result we'll see with our phenotype or the drug testing, it might be the results are coming not only from that single point mutation or the mutation in the patient, but probably from because of the different genetic background. So to overcome this, we have a wonderful technique that is CRISPR-Cas. So now we have a tool that we can actually make edit. We can generate the mutation, or we can correct the mutation. So of course, we can actually we can take the patient line, and then we differentiate, make the patient iPSCs, and then we correct that mutation to make a control line that have similar background, but only the difference is the mutation that is at one point. So we call them isogenic line. And of course, then further you can differentiate and you make the cell type of your interest, and then you can study. So there is this disease, it's called Incontinetia pigmenti. So it's an X-linked disorder, and the gene that have been reported for this is IKBKG. So it's involving an F kappa beta pathway. So basically, it only appear in females, and the female have a multi-system disorder. They have abnormalities in their, they have blindness, they have some neuronal phenotype, brain phenotype, they have epilepsy, and they have some abnormalities in their nail, teeth, and like different body systems are involved. And since it's X-linked, so the female have skewed X inactivation. So it's mean like it's not, the X inactivation is not random. So only the disease one is inactivated. But male cannot show the phenotype because the male with the mutation in this disease, they don't survive. So they die during the early development. So there is a fetal death in the affected male. So there is a no way to study these kind of diseases because you don't see the patient. You don't actually see what the problem patient have, if the patient have a mutation, if the male patient have mutation in this gene. So what we were interested to study this in male. So we actually made a knockout line in a male. So this was the male line and iPSC's line, and then we deleted a portion of IKBK gene that was ultimately a knockout, and there was no protein present. So, and then of course we differentiated into our cell type of interest, and we are doing some analysis, but this is how we are making isogenic lines in lab. So this is the figure that I put here because, so there are like certain challenges, or I would say problem with this system, with the disease modeling. So that is because you are making one cell type. But if you are studying a disease of brain, so brain, have a different cellular population. It have a different cell type. It have, and different cell type interact with the other one. And of course, there are some supporting cells as well. So, and of course, their organization is different. So there are different ways. Now scientists have other ways to study the diseases. So one is the 2D culture. So that is basically you just put the cells in a dish, differentiate into your cell type of your interest. But then we have this, we actually make now organ in lab. So we call, for example, if we make brain organoid, so it's a mini brain in the lab. So you can actually make human brain in the dish. So it's also called disease in a dish, or the brain in a dish. And then now we have another option that we actually can use certain chips, and then on the chip we can put the cells, different type of cells, and then we see that how these cells interact with each other, and there are some other tissues. And now the 4D is actually, it's still not in, there's not studies on this one, but there's, like, I will show you a recent study that you can call somehow 4D, but it's like you put different organs, and then you put their, like you can actually supply them the nutrients, and you can actually see their organization and their interaction. So we'll just look briefly after this. So there's this study where we use the 2D model. So it's a Mowat-Wilson syndrome. So basically, it's a neurodevelopmental disease. So the patient with this one have epilepsy. So even this, if there is this one mutation, like if there is a deletion or a nonsense mutation on one allele, so that is sufficient to cause the disease. And they have heart disease. They have a couple of like epilepsy and other problems. And the mutation in ZEP2 gene, that is a very essentially early in development, so that is reported to cause this disease. So what we did, we took the patient, two patient and two control, we make them into neuron, we differentiate, we made the iPSCs, and then of course we differentiated those iPSCs into um, progenitor cells, and of course we further differentiated into neuron. So this is like how the early progenitor, they 10 cells look like, and these are the neurons. So basically they express the mature neuronal markers, for example MAP2 and GABA. So what we did with the, so if you are interested in disease drug, and you want to see that you are making a drug that affect the brain, a certain neuronal type, so you can make that neuron, you can put the drug on, and you can see like how the drug is affecting. You can do further like functional analysis like electrophysiology and other things. But what we did transcriptomic, we did electrophysiology, and we check like our patient neuron, how they are firing the action potential as compared to our control neuron, and we saw some electrophysiological abnormalities. So I'm not going into detail of these things. I just wanted to give you a brief idea like how we are using this model to study the diseases. But if you are interested, you can ask me a question later and we can discuss more in detail. And there was some migrational abnormalities. We also did transcriptomics, and we see that there was some genes that are involved in DNA binding regulation or transcription, and the forebrain development, they were dysregulated. So then we actually had other genes that were important in the same pathway. So what I was doing in my previous lab, so I made a knockout of other genes like KMT2D gene, and there was POLR2. So the gene that was very prominent in this study. So we wanted to see that, okay, if there are a shear pathway that have not been reported previously, so if we can look on those. So we were doing that kind of work. So that was the 2D model. So as I mentioned earlier, but 2D is just culture, like just in a one layer. But how we can study the brain organization or the organ, any organ, it could be heart, it could be kidney, it could be any. So then the other model that we have is called organoid. So as the name indicates, organoid is basically that now you can make organ. So here is the brain. So here you can look the organization of different cell types. And if you, this is the artificial. So actually you can now, so it's almost show the same thing that we see in the brain in the lab that we artificially make. And there's a lot of, the supporting evidence coming from single cell transcriptomics and other very, different techniques, that what we are making in the lab is actually similar to the human brain, or to some extent. And then the other thing is, now people have started using making different organoid and combining them, and then they see that, okay, how they interact, how, for example, if this is a one part of the brain and this is the other part, and how they actually interact with each other. So now we call them assembloid. So for example, there was this one paper, what they did is actually they made a neuromuscular organoid. So basically they made the organoid, this is again the organoid, so not assembloid. So they have the actually muscle cell, they represent the muscle cell and the brain cell as well. So then they did functional analysis, and they could actually, if you're interested, the link, the reference is there. So it's a very cool way to study neuromuscular diseases. Then this lab in Stanford, I think so. They are pioneer in Assembloid actually. So what they are doing, they are making different regions and they are connecting them, and then they are studying. For example, they study two different regions of the brain, and they could see. So this was their actual picture, so that you could see that these neurons are migrating and inside. So now you can actually dig more deeper into the functional analysis of your organ of interest. So the same lab, they actually had a couple of more papers where they have made assembloid. So they made actually cortical interneuron, and then the neuron and the muscle cell, and they could actually study the neuromuscular junction and things. So this is that was the way of doing the disease modeling. So how we do the development of the drug. So we all know that the drug development is not an easy process. So we start from the basic experiment in the lab. We probably start from, first we see if this compound can actually affect this it can be used as a drug for this certain disease. So we do some experiment in the lab, then we go for, we do experiment on the animal models, mostly the mice model, rabbit, or so, and then we go for clinical trial, like phase 1, phase 2, phase 3. But since again, animal are not human, so of course they cannot fully show the same thing. And there are a couple of evidences that where we have, we know that all diseases cannot be studied in animals. So, and of course, if there is a, the drug, if the drug don't work on the animal, then of course it's a waste of couple of years. Like usually it take more than 10 to 12 years, and of course couple of million US dollar. So what we have now, so we take the cells from the patient. For example, if you are making a disease drug for ALS patient or Alzheimer, so you can now take blood sample or skin sample from the Alzheimer patient, you make iPSCs, you make them into the cell type of interest, and then you can study your compound directly on the patient cell type. And it's usually high throughput. It's, and then you can actually give it another level of complexity, so that we'll see in the next slide, and then you can actually go for the clinical trial. And of course it's a disease specific, it's a genetic related to the genetic background. So it's actually the fast way or the most appropriate way now. And there are a couple of disease for ALS. There's a group working in Harvard. They are working on ALS. They are pioneering iPSCs. And then there is a trial on Parkinson. There is a trial on Alzheimer. So there are a couple of trials on the diseases. For example, for ALS, they are making motor neurons, and then they are studying the drug on the motor neurons. So what here we have, so there are different ways to study. So one is that we make like control and patient, and we study the drug. Then the other way is that, for example, here they make like motor neuron and the muscle, and then they connect those in this chip, and then they see like how they interact, and you can study the drug mechanism and disease mechanism as well. So the other is the one of the main problem in most of the drugs is that they don't cross the blood brain barrier. So if you give the drug, if your drug is if your disease is related to brain, and you are putting the drug in the blood, so how this drug is gonna cross the blood brain barrier and go into the brain. So now they actually making this kind of structure where you have this portion where you put your blood, and then of course it's an artificial way of doing it with the human cells. And this was the one I haven't seen, like this is still a little bit theoretical beside the assembloid, but this is a future way of doing it, like we can put different organ and effective drugs on different organ. So another thing is the cell therapy. So what happened, for example, if the patient have a disease, for example, thalassemia is one of the most common disease in Pakistan as well. So it have 6% carrier frequency, and there are a lot of disorders that cannot be treated. So what scientists have this option is that take any cell type, fibroblast, blood cell, whatever, they make them into iPSCs, similarly putting Yamanaka factor, and then they differentiate into cell type of interest. So if you are treating a heart disease, if you are treating a neuronal disease, if you are treating a muscle disease, or whatever disease you are treating. So the cell that you make can actually study the mechanism of the disease, and then you can use them for drug screening. Both we have studied in our previous slide we discussed. And then you can, for example, if this is coming from a patient and patient have a genetic disease, uh, it have a mutation in SMA gene, it have a mutation in thalassemia, or for any type of genetic disease. So what we have option, we can correct that mutation and we put those cells back into the patient. For example, if we are taking the skin cell and but the patient have a neuronal disease, so we edit those cells and the neurons, and we put back into the patient. And of course, these are the patient cells, so they will not be rejected, so they will not have a problem of using immunosuppressant and those things. But what problem this have is cost. So this is quite a costly technique. So iPSCs I was working in Uppsala University, and we were the only group working on iPSCs in the whole university. Of course, there was a couple of groups that later they started with working with iPSCs, but still it's a very expensive technique. So what other option? So the same lab, Yamanaka's lab, the guy who got Nobel Prize of developing iPSCs, so now they're working on finding certain donors that they call super donors. So what the super donor is, that actually they have the match HLA type. So basically, only certain individual in the whole population can be used for the regenerative medicine, because they will not be immunorejected, so they can be used for the cell therapy. So basically, for whole Japanese population, they came across that the 50 super donor can actually cover the whole Japanese population. So they don't have to make cell line from each individual. So if there is a donor that have the right HLA type, so that can be used for this kind of cell therapy. So we have the normal donor, we make the iPSCs, and then we differentiate. Since it's normal, we don't need to make the disease editing, genome editing, and we put them in back into the patient, and this is the way of treating it. So yeah, we are moving to the end. So basically, end of the presentation, but not the end of this research. It's just the beginning. And if you look that we already have, I mean, here we cannot put all the development, but this is the technique, the first paper where they actually showed the concept of making these type of stem cell that they call induced pluripotent stem cell came in 2006. And then of course they showed later in human, the first was on mice, then human was 2007. And from there afterwards, you can actually see a lot of development, a lot of clinical trial have been started with different type of disorder. And especially now with the addition of CRISPR, now we have even more clinical trial coming, and the drug discovery and the drug commercial iPSC plant. So actually Japan is quite ahead in this technique, because of course USA is also, but since the guy who discovered was from Japan. But with all fancy things I showed you that we can do with iPSCs, of course, we have certain problem. So one is that research and diagnosis and treatments are two different things. So for diagnosis, of course, we need the right line, but for the treatment, we need even more clinical grade lines. So the line that have potential to differentiate into like right cell type, and it give us a good quality of differentiation. And of course, iPSCs that can be used for therapeutic products. And of course, the so the problem with this is, so for example, if you are making neuron and there are still few cells left in that culture that you put into the patient, and that can cause tumor. So we also have to overcome this problem. And of course, if we still we have this super donor thing, but of course we still are not sure like how good that will be in if there is an immune rejection. So we have to work on this. And also cell to cell variation we have to look. And also uh, we have been studying these diseases like mostly monogenic diseases, but now it's time to move towards the polygenic disorders. For example, schizophrenia, that we don't have answers for that kind of genetic part. We know that there are a lot of genes variant, but how those variant and those genes are affecting the function, we have to look on that. And we also need a large patient cohort iPSCs and these kind of. And genetic instability is a very important point. So what actually happens if I'm culturing cells in the lab? So after certain time of culturing, like we call them passaging. So after certain passages, they have this ability to acquire mutation. They have these abilities to make acquire chromosomal abnormalities. So we have to also overcome this. So of course, iPSCs can be used for disease modeling, for the cell therapy, and for the drug discovery. And of course, it can be used for the neurodegenerative diseases and a lot of organ injuries and the disorders that did not had any treatment in past. So now we can actually use this with other techniques. For example, CRISPR we can use for, we can correct the patient mutation, give the patient back. Actually, so now there are a lot of studies. Of course, with the interest of time, I didn't put all of those here, but there are now the cancer therapies using stem cell plus CRISPR. There is already there was a paper in New England Journal of Medicine this December, where they actually use CRISPR to correct the mutation in hematopoietic stem cell to treat thalassemia and sickle cell anemia. So of course, again, the same thing that I have mentioned in my previous slide, that with all the things that we have, all the opportunities or possibilities that we have with this cell type, we have a lot of things that we have to still look up and overcome. So that's it. So of course you can differentiate into any cell type of interest, and I would be very happy to take questions. So I kept this in most like mostly to the educational purpose. I didn't put a lot of my research work detail, but if you are interested, I can talk more about it.
Thank you so much, Dr. Amreen Fatima. It was a really nice presentation, and it was very it has enlightened us upon the topic very well. I would like the participants, if anyone has any question, they can raise hands and come one by one so that Dr. Amreen can answer everyone. So you all are more than welcome.
Yes, student, please ask question. So there's something in the chat.
जी अंकिल थैंक यू वेरी मच
Hadika, it's a very good question. So basically, there is a problem. It requires the mutation. So there are few things. So for example, if I'm working for the research purpose, it happened with me. When I was making my CRISPR line, so I made the line, and it take so the iPSC culture is not an easy culture, because you have to be in lab every day, because you change your media every day. So now there is certain media where you can skip a day, but it's still a very tedious job. So when you make CRISPR knockout, and I made, and ultimately when I looked on the karyotype, so there was they acquired an extra chromosome, or in one there was a deletion. So I couldn't use those lines for my research purpose. So yes, this is a problem that they acquire certain mutations, but then the way to overcome this is that if you are using for the drug purpose, so we have these techniques, so we can actually look after every 5 or 6 passages, after a month or 2 months, we can still look on if the our chromosome are intact, if they are in the right um, form, or we can actually look for some certain markers and see if our lines are still the same iPSCs line. So that is the way to overcome. And if you are working on something like cell therapy, so then you go for even the whole genome sequencing, and you see if they have acquired a mutation. So it's a normal thing that in culture, so your culture require the mutation, and if you look on the other cell type like HeLa, HEC, or even the embryonic stem cell, they have like the similar problem. In fact, the other cell line have more problem than this one. But then of course we can go for whole genome sequencing, again a very expensive thing, but if your purpose is the clinical, then of course you can go for that, or the otherwise we can look on the karyotype and see.
मैम मेरा एक क्वेश्चन था,
जी बोलिए।
बल्कि दो उसके पार्ट हैं क्वेश्चन के। एक तो ये कि हम लैब के अंदर स्टेम सेल को कंट्रोल करते हैं या नहीं करते। दूसरा ये, जब हम अगर इंजेक्ट करते हैं किसी ह्यूमन बॉडी में, तो वो तो स्टेम सेल का प्रॉपर्टी है कि उसने डिवाइड होना है, तो वहाँ पे कैसे कंट्रोल होंगे वो?
ओके, सो अभी हम ये करते हैं कि अगेन देयर आर certain things. So first thing is, then when we the first was that we grow cells in lab. हम लैब में क्या करते हैं, आपका ये था ना कि हम लैब में बना सकते हैं।
जी, मैंने पूछना था कि लैब में जब हम बना रहे हैं उनको, तो वो कल्चर पे डिवाइड हो रहे हैं, मुसलसल उनके नंबर इंक्रीज हो
जी,
रहे हैं, क्या हम इस जो इंक्रीज हो रहे हैं इनको कंट्रोल कर सकते हैं?
अच्छा, हम उसको ये करते हैं क्योंकि उन्होंने उनकी प्रॉपर्टी ही है ना, देयर आर टू प्रॉपर्टीज, सो वन इज दैट दे ग्रो, एंड द अदर इज जब हम उनको डायरेक्ट करते हैं। अब आप ये भी है ना, सो वन प्रॉपर्टी इज दैट दे ग्रो, दे मेक सिमिलर cells of their type. But when they differentiate, so when we give them a direction, we give them a we give them certain transcription factors, so we call them directed differentiation, then they differentiate. But before we do that, we grow them in lab. So what we do usually is we grow and then we put them in cryostore. So we put them in liquid nitrogen and we put them in -80. So usually in liquid nitrogen, and whenever we need, we can regrow them. We can just put them back in the culture. So for example, if I'm interested in a line, in a disease line, for example, autism, I want to study autism spectrum disorder, and there is a group working in Germany, USA, or anywhere. So I can, and they have made the line. So I can request them कि आप मुझे अपनी line भेजें, and even they can send and I can grow the line. So इतना हो सकता है that you can actually freeze them, you can put them, and they can be used for the further purposes. And the other part of question is was that when we put them in the patient, right? This was
Yes.
related to that. So what happened? So for example, what this was one of the challenge. So हमारा जो ultimate aim is that to make a cell type that is उसमें 100% जो cells हैं वो वही हैं जो हमें चाहिए. So what we do actually, if I'm making a cell type and my cell type is not pure, so I actually have this, we can actually sort the cells. Sorting में ये होता है कि हम certain markers use करके, so we can only capture the cell that we are interested and the rest we discard. And then we grow those cells in culture and we see that okay, these are the very homogeneous cell population, then we go for the patient. So, and then of course there are certain things. So one is that you give them cell type that is needed. For example, if there is a heart injury, you give them heart cells. if there is a something neurodegenerative disease, you give them neuron. But for example, in thalassemia case, so of course blood, so aapne blood cells nahi kar sakte, because then you have to redo again and again. So in that case, what we are doing is we can actually put them, give them a cell type that is multipotent. For example, the bone marrow hematopoietic stem cell. So they can give rise to the normal blood cell, or फिर उनका वो जो thalassemia वाला चीज है, they will have a normal oxygen carrying capacity. So we can give the stem cell, or we can give adult cell. We can give both cell type. But again, one thing that we need to make sure is that this is the right and the clean population that we are giving to the patient. Sorry, so there was a question. Okay. Isn't it dangerous to expect the stem cell? No, not really. I mean, now there are groups working on this thing. The only one dangerous part is if we are not giving the right cell to the patient. Then of course it is. But otherwise, if it's like well characterized, and if it's the right population, then definitely that is doable. And people, of course, I haven't put everything, but if you go and study, you can see there are phase 3 clinical trial for the stem cell therapy or this. And there are now people, there are certain things like, for example, the mesenchymal stem cell. This is quite old thing. So we take mesenchymal stem cell from the normal individual, and then of course we give for the knee replacement, or अगर कोई भी bone related disease होती है तो उसके लिए हम देते हैं. So here I have a question. When do I think Pakistan has? Yes. So here are your first question. If the Pakistan have a setup, I'm not sure about the rest of the universities, but we do have a setup at AKU. So I just joined recently, but there is a Center for Regenerative Medicine and Stem Cell Research. So they are working on. So there is one person. She's also working on iPSCs. But I have, since I joined just recently, so I am, my interest is in disease modeling, in iPS disease modeling. So my plan is to start disease modeling here at AKU. And the second was that, yes, we can definitely do the mutation reversal. We use the CRISPR. So I'm sure you all are familiar with the CRISPR. So with the CRISPR, we can knock out the gene, we can remove the gene, we can insert the gene, we can make the mutation, we can correct, or even we can correct the mutation. So it work on two different mechanisms. So one is this non-homologous and joining, the other is homology directed repair. So we provide a piece of instruction. So which kind of mutation we want, or we can provide a piece of instruction that is in form of base pair oligonucleotide, and then we can instead of the disease one, we provide the instruct that have the right the nucleotide, and that can be replaced. So I have worked on CRISPR, like using making iPSC in isogenic line, or of course correcting the mutation and generating the mutation. So yes, we can do, and same is happening with a lot of stem cell therapies.
अच्छा डॉक्टर अमरीन, अगर हमारे को स्टूडेंट्स स्टेम सेल्स पे काम करना चाहते हैं तो आप उनको क्या सजेस्ट करेंगी, कैसे गाइड करेंगी, अगर आप थोड़ा सा हमें बता दें इसके बारे में.
अगर स्टूडेंट्स मुझे बताएंगे कि वो क्या करना चाहते हैं, देखें, again the thing that I have mentioned, that this is a very very elegant science, and I mean, if you look on the latest trend, now we have a lot of techniques. We have a single cell. हम एक, अगर आपके पास एक, for example, you have a section of brain, so we can actually now look on each cell individually. कि उस cell में, for example, we know that we have 20 to 25,000 genes, so we can check the expression of all those genes in one cell. Not only the expression, we can look on the proteomics. We can actually look on what protein I'm expressing. We can look on chromatin modeling and everything. So there's a lot of things that are coupled with the stem cell, but still it's one of the most, um, I would say, important work that we, because it's exactly related to human and with a lot of promise. Okay, so now it comes with the cost, and we know that this is very costly, but we I would suggest that if you are a master student or you are doing your undergrads, so first thing I would like, if you have this opportunity of going abroad, doing your masters abroad, and even the PhD abroad, so you can, one is that you join a lab that is working on that thing. So in Pakistan, of course, we don't have the setup, but since I would be working, so if there is a like small kind of internship or those kind of things that we can arrange probably in near future, inshallah. But मतलब रिसर्च के पर्पस के लिए अगर आपने हार्ड कोर रिसर्च करनी है इस चीज में, तो फिर हमें यही है कि वी हैव टू गो अब्रॉड और वहाँ पे हम करें, पीएचडी करें इसमें, एंड देन फर्दर पोस्ट डॉक, and then you can go to the companies. So PhD करने के बाद आप, because now a lot of companies are doing this kind of work, or even blood brain barrier, or even there are PhDs from the companies. So companies like stem cell, Roche, they offer PhD. So आप वहाँ apply करें, masters के लिए करें, तो यही हो सकता है. Any more questions? I hope I answered all, but if you have, please unmute yourself and uh, just ask your question.
I think Dr. Breen, there are no more questions. Thank you very much for your
There's this one question
valuable
from
time.
Imran. So the treatment using my own stem cell safe? Uh, yes, it is. And of course, the whole idea of using your own stem cell is that uh, your immune system will not reject, because that the cell would be part of your body. But then, of course, we have to look like which disease we want to treat, and how how good our differentiation protocols are. So अब हमारे पास एक tool आ गया है, but it doesn't mean that we have to we can do for everything, but we have to look on the clinical grade कि जो हम कर रहे हैं वो ठीक है ना. I mean, it doesn't, instead of treating a disease, it shouldn't cause another disease. So ये वाली चीजें हमें देखनी होती हैं. So that's all.
Thank you so much. Thank you so much, Dr. Naveen Fatima. It was a really good session and it was very informative. And I hope all the questions are sorted by now. Um, I would like to request any of the hosts to end the meeting after. If anyone has any remarks or anyone has any comments, they are more than welcome. And thank you so much again for your presence.
Thank you, Dr. Fatma and all the organizers for inviting me, and thank you. And you are most welcome to ask me. I just put my email, and I would be happy to guide you, or if there could be anything. And good luck.
Okay, thank you, Doctor.
Thank you so much.
Allah
Allah
Hafiz.
Hafiz.